I'm actually going to dead second, I'll take you back outside. Adenoma?
Yeah, so that's what I was trying to do. It was kind of
I'll do my best. Probably too much information. Clock, so I think we'll go ahead and get started. It's really my privilege today to introduce Dr. Andre Ballesteros-Tato from the University of Alabama in Birmingham for a seminar. Andre received his PhD in molecular biology from Autónoma University in Madrid, Spain. I just figured out how to pronounce that. From there, he actually joined Troy Randall's lab at the Trudeau Institute, where he was there for about 6 months, from what I can gather, before going to University of Rochester for about 4 years, and then transitioning to UAB after that. He said that was a very good move for him, moving from the somewhat colder climate in University of Rochester down to UAB. So at UAB, since 2015, he's been an assistant professor in the medicine in the Division of Clinical Immunology and Rheumatology. He's really made a lot of important contributions. He's going to talk about many of those today in the field of immune response to influenza infection, particularly in the area of TFH cell function, and also the role in generating antibody-mediated responses. He's published a lot of in top journals, including Nature Immunology, Immunity, Nature Communications, as well as Journal of Experimental Medicine. He's also received early career investigator awards from the AI, the Biogen IDEC Foundation, and then also a Pittman Scholar Award from UAB. In addition, he serves on the editorial board of Scientific Reports, and he's going to tell us about IL-2 and regulating TFH cell responses. So please join me in welcoming Andre.
Okay, can you hear me? Right, thanks Ken for the introduction. It's a pleasure to be here, and thanks everybody for coming this morning and for taking the time to meet me. I'm really having a great time here. So today I'm going to talk about a particular subset of immune cells that we call T follicular regulatory cells, and I'm going to refer to them as TFRs from the rest of the talk. And I'm going to talk about how the T follicular regulatory cell response is dynamically controlled by IL-2 following influenza infection. And I'm going to show you what these cells do to the B cell response to influenza. So I have divided my talk into two main parts. So in part one, I'm going to be talking about how dynamic changes in IL-2 controls the TFR response in the context of influenza virus infection in mice. And in the part two, I'm going to talk a little bit about the role of these cells in controlling the B cell response to influenza virus. But before going there, I'm going to spend a few minutes talking about T follicular helper cells, germinal center B cells, and regulatory T cells. And hopefully, this will help us to understand why we care about these T follicular regulatory cells. So when you think about the immune response, one of the more important events that take place is the activation of the Cd4 T cells. And the Cd4 T cells are important because they produce pro-inflammatory cytokines. And these pro-inflammatory cytokines produced by the Cd4 T cells help the activity of other immune cells, like, for example, CD8 T cells, to fight the infections by the pathogens, virus, bacteria, also tumors. So they are quite important, the CD4 T cells, because they help other immune cells. And the initiation or the activation of the CD4 T cell response usually happens very quickly after infection. Usually it takes in between hours to 1 or 2 days, depending on the type of infection that you are studying. And it's initiated by the interaction of CD4 T cells with antigen-virulent dendritic cells. So what's happening here is that the dendritic cells are immune cells that are usually in the target tissues where the infection happens. And the dendritic cells take antigens, get activated by the pathogen, take the antigens, migrate into the secondary lymphoid organs where the Cd4 T cells are. And once they encounter the T cells, the dendritic cells are able to activate the T cells by telling them, hey, something is going on in the tissue, you have to go there because we have a pathogen. And when this happens, when the naive cells recognize the antigen and receive costimulatory signals by interacting with the dendritic cells, the naive T cells proliferate. They get activated and proliferate. And then during this proliferation, they differentiate into what we call effector subsets. And those are Cd4 T cells that produce pro-inflammatory cytokines. And as I told you, the pro-inflammatory cytokines help other immune cells to recruit more immune cells to the effector site. And depending on the context and the type of infection, there is basically three big groups of or kinds of effector CD4 T cell responses. We have Th1 cells, which produce mainly interferon gamma, Th2 cells, which produce a lot of IL-4, IL-5, or IL-13, and then we have Th17 responses, which produce IL-17. Th1 cells help to fight infections against intracellular pathogens like virus. Th2 cells are very effective on helping immune response against extracellular pathogens, for example, nematodes. And Th17 response are important for fungal infections or extracellular bacteria. And the differentiation into each of these subsets is controlled by what we call master transcription factors. For example, Th1 differentiation is controlled by Tbet, whereas Th2 differentiation is controlled by GATA3, and Th17 differentiation is controlled by RO-gamma. So at the same time that some of the naive cells, when they interact with dendritic cells, differentiate into the effector pathway, some of the naive CD4 T cells upregulate a transcription factor named BCL6. And when they do that, differentiate into what we call T follicular helper cells, or TFHs. And what is special about these cells is that these cells express high levels of a chemokine receptor named CXCR5. And the expression of CXCR5 allows the T follicular helper cells to migrate from the T cell area. So when you think about the secondary lymphoid organs, you can divide them into two big areas. The T cell area, where all the T cells hang up, and then the B cell areas, which is where the B cells are. And the T follicular helper cells, because express CXCR5, can migrate from the T cell zone into the B cell area. And once they get into the B cell follicles, the T follicular helper cells can interact with germinal center B cells. And during these interactions, T follicular helper cells produce a lot of CD4 T ligand and IL-21 that help the B cells in the germinal centers to undergo a series of changes, including clonal expansion, somatic hypermutation, and affinity modulation. And what this does is that the germinal center B cells that interact with the T follicular helper cells finally differentiate into memory B cells and long-lived plasma cells. And what is special about these cells is that they produce high-affinity antibodies for long periods of time. And this is important because it's the production of this long-lived high-affinity antibody response what confers protection, for example, after vaccination. So it's important that we understand how the T follicular helper cells, how the germinal centers are regulated, because then we can ideally promote or boost vaccination strategies. So as I told you, when T follicular helper cells get into the follicles, they interact with the germinal center B cells. And in this context, if the germinal center B cells and the T follicular helper cells are specific for vaccines, tumors, or pathogens, that's great, because leads to the generation of high-affinity memory B cells and plasma cells, which produce high-affinity antibodies, and you get protection. But there is a dark side of the germinal center response, and it's that sometimes you get T follicular helper cells and germinal center B cells that instead of recognizing foreign antigens, meaning bacterial or tumor antigens, they recognize antigens that are expressed on the cell surface of your own cells. For example, think about collagen or nuclear antigens, DNA, histones. And when this happens, these self-reacting T follicular helper cells and self-reactive germinal center B cells produce autoreactive memory B cells and plasma cells. And in that case, they produce high-affinity antibodies, but against your own antigens. And that's bad because it leads to the development of antibody-mediated immunopathology. and as in the long run, autoimmune disease development. For example, think about lupus, rheumatoid arthritis, or any other antibody-mediated autoimmune disorder. So it's important that we understand how these responses are regulated, because we can either promote this response to vaccines, pathogens, and tumors to get more protective immunity, or if we are working in the context of autoimmune disease development, we can try to prevent the self-reactive germinal center responses to prevent self-reactive antibody responses and therefore prevent autoimmune disease development. So an interesting event that happens when the T follicular helper cells interact with the germinal center B cells in the B cell follicles is that the germinal center B cells undergo a process known as somatic hypermutation. And what this is that the BCR is the antigen receptor that the B cells express and is what they use to recognize the pathogens. The BCRs start to mutate. And during this mutation, the cells that better recognize the antigen are selected. So it's a way to increase the affinity of the B cells for the pathogens. And that's good because it's what it leads you to the generation of high affinity antibodies. The problem with that is that somatic hypermutation is a random process. So when the B cells start to mutate the BCR, there is a risk of generating autoreactive B cell clones. So what this means is that accidentally, when these cells mutate, some of the clones might now recognize self-antigens with high affinity. And if that happens, it might lead to the generation of autoantibody secreting cells. These are B cells that have been generated during somatic hypermutation, and now instead of recognizing pathogen antigens, recognize self-antigens, and that leads to autoimmune disorders. So we know that there must be mechanisms that prevents the generation of autoreactive B cells in the germinal centers, but we don't know which mechanisms are those.
So, can I ask a quick
Yep,
question?
sure.
My ignorance here. So, are autoimmune disorders basically random events?
So,
in the individual?
So autoimmune disorders are, that's a tricky question because we know that when you have an infection, some infections are actually triggering autoimmune diseases. And we know, for example, that some immunological insults like a bacterial infection can be the triggering of autoimmune disorders. And that sense, it may be a random disorder, meaning that it generates from an infection that leads to somatic hypermutation, and then you get accidentally your B cell clones, and because you have an inflammatory environment, those B cell clones can actually move forward and develop into plasma cells.
So another way I guess I might ask you is, then is the more infections one encounters, the greater one's chance for developing an autoimmune disorder just because of the linear random?
That's true and not at the same time. So this doesn't usually happen because there are mechanisms to prevent that. But then for some people, when you get infected, certain patients, they have more chances to get autoreactive B cells because some of those mechanisms doesn't work. So there has to be a background with a deficiency or something that promotes that. So it's not like because you are getting infected, you're going to develop autoimmune disease development, but you have a chance if you have another defects. So to summarize this, the germinal center is good because it can lead you to protection. That's the good part of the germinal center response. But also it can happen that it's responsible for autoimmune disease development. It will be the dark side of the germinal center response. And I want you to remember that you can easily go from here to here because you get infected. But it has to be mechanisms that prevent you from going from here to here to here. Okay. Another immune cell that I want to talk about today is regulatory T cells. So the regulatory T cells is a subset of CD4 positive T cells. So when you think about T cells, you have CD8 T cells, and then you have CD4 T cells, and there is a bunch of different subsets of CD4 T cells. The Tregs, or the regulatory T cells, is one of these subsets. And they are characterized because they express high levels of FOXP3. And this is a transcription factor. And what these cells do is they secrete anti-inflammatory cytokines. And they have, because of that, they have the ability to suppress ongoing T and B cell responses. And actually, they are quite important for maintaining what is called immune tolerance, because we know that in humans and in mice, if Tregs do not properly develop, then mice and humans develop a catastrophic autoimmune disorder. So they are actually fundamental to maintaining the immune system in check. We're getting now to the T follicular regulatory cells. So in 2011, 2 papers came out back-to-back in Nature Medicine from Carola Vinuesa's group and Chendon group. And what they saw in these papers is that some of these regulatory T cells upregulated BCL6 and CXCR5 and differentiate into T follicular regulatory cells. So if you remember from the beginning, we have T follicular helper cells that express BCL6 and CXCR5, and they go to the B cell follicles and they help germinal center responses. Well, these regulatory T cells that express BCL6 and CXCR5, they also go to the B cell follicles, and once they get there, what they do is not so clear, but we know that they might have the ability to suppress germinal center and ongoing T follicular helper cell responses. Because they are regulatory T cells, they secrete from anti-inflammatory cytokines, so we know that Tregs suppress immune responses. Those guys are just Tregs that have the ability to migrate into B cell follicles, so it's likely that they also suppress B cell responses when they get there. So if you think about that, now we have a subset of regulatory T cells with the ability to go into the B cell follicles. So it's likely that the factors that control the differentiation of T cells are going to be important for how the germinal center and the T follicular helper cell response are regulated in vivo. However, we don't know. We know very little about these cells, and we don't know which factors control the formation of T follicular regulatory cells. And we don't know what's the exact role of these cells in the germinal center reaction. And this is what I'm going to try to show you today. So we were interested in these cells and what they do, how are they generated, what mechanisms regulate their response. And to answer all these questions, we use an influenza virus infection model. This is a virus, we use a virus that infects mice. And so we took B6 mice, those are wild-type animals, and then we intranasally infected them with an influenza virus. And that, when we do that, then the mice get sick, they start losing weight. By day 5, 7, the virus is gone, and they start recovering. So it's pretty much the same thing that happened to us when we get infected with flu. We get sick for a week, and then we start recovering. And then we infect the mice, and then at different time points, we analyze the kinetic of expansion of the T follicular helper cell response, the germinal center B cell response, and the kinetic of expansion of our T follicular regulatory cells. And this is the kinetic of expansion of the T follicular helper cells. And so what we do here, so here you have the frequency of BCL6 CXCR5 expressing cells within the CD4 positive T cell population. And you see here that we have some cells that express BCL6 and CXCR5. Those are the T follicular helper cells. So here you have the frequency at different times after infection, and down here you have the numbers. And as you can see here, when we infect the mice with influenza, we have an expansion of T follicular helper cells. The frequency and the numbers of T follicular helper cells peak between day 7 and 15, and then we have a contraction. And this is pretty much very similar to other kinetic of T cells. You get the virus replicate, the antigen gets into the lymph node, T cells recognize it, they start to proliferate, the virus is eliminated, and then you no longer need T cells, and there is a contraction, and some of the cells survive as memory cells. So we analyzed the kinetic of expansion of the germinal center B cells. So we use flow cytometry to detect germinal center B cells, and very briefly, we gate on B cells, and then the B cells that express FAS and PNA, those are the germinal centers. And as you have here the frequency of germinal centers within the B cell population, and here you have the numbers. And once again, we have a quick expansion of germinal centers following infection. We have a peak around day 15, and then there is a contraction, and then some of the germinal centers survive for more than 100 days. This is expected based on the kinetic of the viral replication. The virus is gone by day 7 and 8, so we don't longer need to maintain immune responses from day 7, and then the immune response is contracted. Okay, so we then analyzed the kinetic of expansion of the T follicular regulatory cells. And this is how we identify T follicular regulatory cells in the lymph nodes. All the data that I'm showing you is analyzing the lung draining lymph node. This is the lymph node that drains the lungs. So here you have FOXP3 expressing cells. Those are the regulatory T cells. And then this is CD69 expression. CD69 is an indication of activation. So if you get on FOXP3 cells that are CD69 positive, those will be these cells. Those are the activated Tregs. Those are the Tregs that are responding to the infection. And within that population, you have some Tregs that express CXCR5 and BCL6, about 7%. This is day 30. Those are the TFRs. And as you see here, those cells also express PD-1, which is another marker of TFRs, and express high levels of GL7, which is an indication of cells, indicates cells that are in the germinal center. So we call TFRs our activated Tregs that are positive for BCL6, CXCR5, PD-1, and GL7. And here's how they behave throughout the infection. And when we saw this result, that was quite surprising because anything else that we look at, there is always a peak in between the 7 and 15, and then there's a contraction, no matter what you look at. But in this case, it's different. If you see here, these are the frequency, and this is the numbers. We detect very few TFRs at the peak of the infection. Where everybody else is expanding, we don't see too many of them. And we have to wait until about day 30 to start seeing a convincing population of TFRs. And as you can see here, those are the numbers. We have very few at day 10, and then they largely accumulate later on. So this kinetic of expansion is totally different from the kinetic of expansion of any other immune cell, meaning that it takes longer to differentiate. So this data suggests that TFRs fail to differentiate at the peak of the influenza infection.
Andre, if you plot actual antibody that's produced, is it in agreement with the germinal center B cell response, antibody produced against the infection?
So the antibody response, you get antibodies that are coming from the germinal center and the antibodies that are coming from the extrafollicular responses. So when you infect mice, you get a boost in antibodies. There is the half-life of the antibodies is about 4 weeks, and then the antibodies decline a little bit, but you're still having a lot of antibodies because you generate long-lived plasma cells which go to the bone marrow and they keep producing antibodies. So it's not, you don't get that big contraction. There is a contraction, but it's still producing a production of antibodies. So I told you that this is good because this is flow data, but I told you that in order to call a cell Treg TFR, they have to be in the B cell follicle. So to confirm our flow cytometry data, we infect mice with the influenza, and then at day 10 and 30, we look for Tregs in the B cell follicles by immunohistochemistry. What you have here on the left, this is a picture of the lung draining lymph node at day 10 after infection, and this is day 30. And in green, you have IgD, which indicates where the B cells are. So everything that you see that is green is a B cell follicle. Then we have CD35 here. This is a marker for the follicular dendritic cell network. This is a surrogate marker for germinal center. So green area is the B cell follicle. Here you have your germinal centers. And in white, you have the regulatory T cells. And then if you look at day 10, you see that we have very few regulatory T cells in the B cell follicles and almost none inside the germinal center. And what's happened at day 30 is that now Tregs colonize the B cell follicles and go inside the germinal center. So these results confirm what we have found using flow cytometry and indicates that TFRs do not differentiate early after infection, and yet you have to wait until at least day 30 to see a convincing population of TFRs. So to summarize this section, I showed you that TFRs fail to differentiate at the peak of infection. However, once the immune system, once the immune response contracts, some of the Tregs upregulate BCL6 and CXCR5, and those cells migrate into the B cell follicles. And as a consequence, TFR cells accumulate later on in the lung draining lymph node. So the obvious question that we wanted to answer is what is going on here? Why we don't have TFRs at the peak of the infection? What is the mechanism that is preventing TFR differentiation? So we look a little bit closer to the phenotype of the regulatory T cells. And one of the hallmarks of regulatory T cells is that they express high levels of CD25. And CD25 is the alpha chain of the high affinity IL-2 receptor. And usually cells that express high levels of CD25 are cells that are responding to IL-2. And Tregs are usually CD25 express high levels of CD25 because they require IL-2 for their function. And what we found is that this is day 30 after infection. Here you have our regulatory T cells, and we actually found that the majority of the Tregs were CD25 high, but some of them were actually quite low. They expressed lower levels of CD25. And when we look for TFR markers in these two subsets, we found that we pretty much didn't find any BCL6, CXCR5 expressing cell within the CD25 positive subset, whereas around 16% of the Tregs that were CD25 low expressed BCL6 and CXCR5. Thus suggesting that unlike conventional Tregs, which are typically CD25 positive, TFRs express low levels of the alpha chains of the IL-2 receptor. So we wanted to understand a little bit more about how these cells were generated. And for doing that, we did this experiment. In this experiment, we sort Tregs from FOXP3 YFP mice, and we sort CD25 expressing Tregs. So what the conventional Tregs, they express FOXP3, and they are CD25 positive. Then we took our sorted Tregs and we transferred them back into mice that don't have T cells. And then we transfer also, together with the Tregs, we transfer CD4s and CD8 T cells to get a competent in a normal T cell environment. Then we infect the mice with influenza, and then we analyze the progeny that derive from these cells at day 30. And we can do that because these cells express CD45.2. So we can track them through the infection. So we infect the mice and wait 30 days. Then we analyze, we gate on FOXP3 positive cells. We have two populations. This is the population of cells that express CD45.2. So these cells are FOXP3 positive and derive from the CD25 high expressing cell that we just transferred. And as you can see here, originally these cells were CD25 high. But after infection, some of them downregulated CD25. And only those that downregulated CD25 upregulated BCL6 and CXCR5. So if you think about a little bit about this, the cells that normally express CD25 are receiving IL-2 signaling. And here we found that only the Tregs that downregulated CD25 are able to upregulate BCL6 and CXCR5. So this data suggests that there might be a negative relationship between IL-2 signaling and TFR differentiation, because you cannot get BCL6 and CXCR5 in the cells that are CD25 high. And this makes sense because we know that IL-2 and signaling through the IL-2 receptor inhibits BCL6 expression. Yeah, go ahead.
I just want to make sure I understand your design here. So you took wild-type endogenous FOXP3 positive cells, transferred them into Cd4 T cell deficient mice.
Mm-hmm.
How are the mice alive?
So we transfer back, together with the Tregs, we transfer back Y-type CD4 and CD8 T cells from Y-type B6 mice. So we transfer our Tregs plus a normal population of T cells to have T cells. But the difference is that the Tregs that we transfer here, they express CD45.2, and all of the other T cells express CD45.1. So we can actually track these cells.
And is it possible to interpret your data as FOXP3 expression requires a B cell to present the antigen reciprocity?
So can you repeat the question?
So is it possible that the antigen presentation by B cell drives the FOXP3?
So I'm not going to show you this data, but we have done similar experiment to this by transferring Tregs, so FOXP3 expressing cells, and also CD4 T cells that do not express FOXP3. And what we found is that if you transfer T cells that are not Tregs, they don't upregulate FOXP3 and they don't differentiate into TFRs. What this shows is that the precursors of the TFRs are actually pre-existing Tregs they express FOXP3 before becoming TFRs. You don't get the other way around.
I'm not sure exactly how you do this, but do you see any evidence that those cells lose FOXP3 expression over time once they downregulate CD25?
So when you compare how they still FOXP3 positive, but compared to conventional Tregs, they express lower levels of FOXP3,
okay.
but still positive. I mean, you can. I don't know if I showed you. I think I showed you here.
Well, I saw it kind of.
Yeah, it drops a little bit, but
back a little bit.
still quite high.
Okay.
So I told you, it makes sense that IL-2 because IL-2 prevents BCL6 expression, and we know that BCL6 expression is required for TFR differentiation. So I show you that at the peak of infection we don't have TFRs, and we know that IL-2 is one of the cytokines that is being largely produced by effector T cells at the peak of infection. So the next question was, well, maybe we don't have TFRs at the peak of infection because we have a high IL-2 environment. And so the next experiment that we did is we infect mice with influenza, and then we wait until day 20, and then we start treating them with recombinant IL-2. And we treat them once a day for 10 days. And what we are doing here is now we are creating a high IL-2 environment by giving them back IL-2, and then we ask whether or not we can have TFRs. Remember, normally at day 30 we have a nice population of TFRs. Now we are giving them IL-2, and we are asking if we have TFRs or not. And as you can see here, this is our control group. They just received PBS, and about 10% of the regulatory T cells express BCL6 and CXCR5. However, this is our group where the mice were treated with recombinant IL-2, and we found only about 2%. And the numbers actually are here. This is day 30 mice. This is day 30 mice treated with IL-2. So it shows that TFRs cannot differentiate in a high IL-2 environment. So we ask, well, now that we know that TFRs cannot differentiate in a high IL-2 environment, how is the IL-2 environment at the peak of the infection? And to study this, we use IL-2 reporter mice. In these mice, the cells that express IL-2 also express GFP, and we can detect them by flow cytometry. So we infect the IL-2 reporter mice with influenza, and then we look at the frequency of IL-2 producing cells at different time points. And as you can see here, we have the frequency of IL-2 producing cells peak about day 10. And then there is a contraction, which makes sense because we know that the effector cells, I showed you that there is a contraction on the effector response. So yeah, IL-2 is highly produced at the peak of the influenza infection. We then infect mice with influenza, and then at day 3 we start treating them with anti-IL-2 antibodies. And what this does is that it neutralizes the IL-2. So now we have mice that are at the peak of the infection will have a low IL-2 environment because we are neutralizing IL-2. And we ask whether or not we now can detect TFRs. And this is the result of the experiment. As you see, as expected, we have very few TFRs in the control group. This is day 10 after infection. This is normal because we know that TFRs do not develop at this time. However, the frequency and the number of TFRs were increased in the mice that were treated with anti-IL-2, suggesting that the increased levels of IL-2 at the peak of the infection is preventing TFR differentiation. To further prove this conclusion, we sort regulatory T cells, CD25 positive Tregs, from FOXP3 GFP reporter mice at day 7 after infection. Then we took the Tregs, we activated in vitro with CD3 and CD28 in the presence of high or low IL-2 conditions. And then we wait 72 hours and we ask whether we have TFRs or not in the different conditions. And as you can see here, in gray you have the high IL-2 conditions. The cells, the Tregs that were activated in the presence of high IL-2, they express high levels of CD25, whereas the other ones express lower levels of CD25. Then we look for BCL6. BCL6 is expressed on TFRs, and as you can see here, we have a lower BCL6 expression in the cells that were incubated in the high IL-2 conditions. And same thing for CXCR5. The cells that were incubated with high doses of IL-2 express low levels of CXCR5. Another transcription factor that is important for BCL6 function and expression is BLIMP. Usually, cells that express high levels of BCL6 are BLIMP negative or low. And as you can see here, when we culture our Tregs in a high IL-2 condition, they express high levels of BLIMP, which we know antagonize BCL6 expression. And as a consequence of all these changes, when we look at the frequency of TFR-like cells, meaning BCL6 expressing CXCR5 cells, the frequency of those cells was much higher in the low IL-2 conditions. Showing that, once again, that IL-2 prevents BCL6 expression and TFR differentiation. So what we think is happening is that when we fed mice with influenza, the TFRs, it takes a while to have TFRs. And it takes a while because the TFRs derive from the CD25 expressing Tregs. But because we have buccal loads of IL-2 at the peak of the infection, this IL-2 is being taken by the Tregs, and the signal, when they take Tregs and they take IL-2 signals upregulates BLIMP and prevents BCL6 expression, which prevents TFR differentiation. Once the immune response is contracting, then the IL-2 levels decline, and then some of the Tregs then regulate CD25 and differentiate into TFRs. So we were wondering what is the mechanism by which IL-2 prevents TFR differentiation. And we know that there are two main mechanisms by which IL-2 can prevent BCL6 expression and activity. One is IL-2 induction of BLIMP, and BLIMP is a transcription factor that prevents BCL6 expression. So it's possible that at the peak of infection, Tregs responding to IL-2 upregulate BLIMP, and BLIMP prevents BCL6 expression. The other option is that IL-2, and we know that because Ken published it in Nature Immunology, IL-2 favors the formation of TBET BCL6 complexes, and when this happens, TBET blocks the DNA binding domain of BCL6. So BCL6 cannot bind to its target genes, and then that prevents the BCL6 biological activity. So we were asking what is the role of TBET and BLIMP in TFR biology. And to answer these questions, we first, here we're going to analyze the role of TBET. And what we did is we did bone marrow chimeras. So what is this? We took TCR knockout mice. They don't have T cells. We irradiate them. And when we do that, all the immune cell precursors are gone. And then we transfer them back, a mix of bone marrow that was 50% from the B6 wild-type mouse and the other 50% from TBET knockout mice. And we can differentiate it because the TBET knockout mice were CD45.1 negative and the other cells were CD45 positive. So what's happened is that if we wait 2 months, then we analyze the Treg response, and as you can see here, 50% of the Tregs are derived from the B6 bone marrow, and therefore are wild type, whereas the other 50% of the Tregs are derived from the TBET knockout bone marrow, and therefore do not express TBET, and we can differentiate them based on the expression of CD45.1. So we generate these bone marrow chimeras, we wait 2 months for the reconstitution, and then we infect them with flu, and then we analyze the B6 and the TBET response at day 12 after infection. So the idea here is that we know that IL-2 at the peak of infection prevents TFR development, and we are asking whether this is by a mechanism that is mediated by TBET. So what we expect to see is that if TBET is preventing TFR development, by taking out TBET, we should see now TFRs at the peak of the infection. But this is not what we found. So here you have the frequency of BCL6 and CXCR5 expressing cells within the wild type, FOXP3, CD25 low population, and here is the frequency within the TBET knockout. So they don't express TBET. And as you see here, the frequency is quite similar, suggesting that TBET does not have a role on TFR differentiation. Otherwise, we should see a difference. We then repeat the experiment, but in this case, we use B6 as a donors and BLIMP flux LCK3 mice. So in these mice, T cells do not express BLIMP. And again, if we create these bone marrow chimeras and wait 2 months, the regulatory T cell population is a mix of wild type and BLIMP knockout T cells. So we infect the mice with influenza, wait 12 days, again, the peak of infection, no TFRs in the wild types, and then we ask whether or not now we have TFRs if the T cells are BLIMP knockout. And as you see here, we have 3% of TFRs in the B6 compartment, but almost 20% of the TFR of the Tregs in the BLIMP knockout compartment express now BCL6 and CXCR5 markers, suggesting that BLIMP expression prevents the development of TFRs. Therefore, if we take it out, we have TFRs even at the peak of the infection. So I show you that BLIM prevents TFR development at the peak of the infection, and in the previous experiments, I show you that IL-2 at the peak of the infection prevents TFR development. So now we wanted to tie up these two events. And what we did is we generate our mixed B6 BLIM knockout chimeras, we infect them with influenza, and then at day 20, we start treating them with recombinant IL-2 to generate this high IL-2 environment that we know prevents TFR development. And here you have, on the upper panels, this is the B6 and BLIM knockout TFR populations in the control mice that were injected with just PBS. And here are the frequency of TFRs in the mice that received IL-2. And as you can see here, if the Tregs are wild type, when we treat with IL-2, we have a reduction in the frequency of TFRs. But if the T cells are BLIM knockout and we treat them with IL-2, the frequency of TFRs is quite exactly the same. Basically, if the T cells do not express BLIM, they don't care about IL-2 treatment. It suggests that IL-2 prevents TFR development by a BLIM-1 dependent mechanism. So here is what we think is happening. And this is the summary for the part 1. We infect mice with influenza. We have an expansion of the effector T cell response, which they start producing cytokines, which help other immune cells, because now the primary goal is to fight the infection. So we need overall activation of the immune system. This, and among the cytokines that are reproduced, cytokine is one of them, and we know the IL-2 is fundamental for the activity of other immune cells, including, for example, CD8 T cells. But as a consequence, we have a high IL-2 environment at the peak of the infection. And that high IL-2 environment prevents TFR development. Once the effector response contracts, the IL-2 levels decline, and some of the Tregs that were activated start to lose C25 expression. And if they receive the right signals, some of those cells will now downregulate BLIM and now can start to upregulate BCL6. When they do it, they migrate into the B cell follicles. Yep.
In those experiments where you're modifying BLIM for IL-2 and you have the TFR population earlier, does that have an effect on the infection or the course of infection or how quickly they clear the virus?
I'm going to show you some data now. But the data that I'm going to show you shows what's happened when you take out TFRs later on. We don't have data to show what's happening if now you have TFRs early on. But that's a pretty good, interesting question. What we think it will happen, we think that this is an active mechanism to actually prevent TFRs from differentiating early on after infection. Because when you get infected, you need to generate an immune response as quick as possible. And an important component of this immune response is the T-dependent and T-independent antibody production. So we think that IL-2 prevents TFR development so that you can quickly generate these broadly antibody responses. And so if you have TFRs at the beginning of the infection, that will prevent the generation of this early wave of antibody responses, and that's not good. Because we know that if we have mice in the lab that do not produce antibodies, they have B cells but do not produce antibodies, and what's happened is that the doses of flu that are normally sublethal, they are lethal for those mice. But we didn't do the experiments to prove that if you have TFRs, then you get less antibody responses, but that will be my prediction. But we did experiments to actually interrogate what's happened, what is the role of TFRs when they accumulate later on, right? And again, this is a picture of a long-draining lymph node 30 days after infection. And I use this to show you again that this is the B cell follicle, and you have some germinal centers here. And as you can see here, when we look at Tregs are inside the B cell follicle, inside the germinal center, and surrounding the germinal center. So the question is what they are doing there, right? The virus is gone, so what's the function? Do they have a function there? Maybe they are helping to contract the germinal center response. And we know that makes sense because TFRs are Tregs, and there are a bunch of papers that show that when you take them out in vitro, they can suppress B and T follicular helper cell responses. So we ask, what are they doing there? And to answer this question, we cross BCL6 flox mice to FOXP3 Cre mice. So that way we generate BCL6 flox, FOXP3 Cre mice in which the zinc finger domains of BCL6 are deleted in FOXP3. What that means is that FOXP3 cannot express functional BCL6. And since we know BCL6 is required for TFR differentiation, it's likely that these cells will not develop a TFR response. So we generated these mice, then we infect them then with influenza, and we look at day 30, and we look for TFRs, germinal centers, and T follicular helper cells. And this is the frequency of TFRs in wild type, and then our BCL6 flux mice. And as expected, we didn't find TFRs, this is the frequency and this is the numbers, in the mice in which Tregs cannot express BCL6. This is what our prediction was. And then we asked what's happened to the TFH and the germinal center response. And here you have the germinal center B cells. We define, in this case, germinal center as B cells that are CD38 low and express high levels of FASH. And as you see here, both the frequency and the numbers were quite similar, whether or not the mice had TFRs or not. And that was quite surprising. We looked for TFHs, and once again, we found that the frequency of TFHs and actually the number of TFHs were similar in B6 and BCL6 flux FOXP3 cream mice, suggesting that the lack of TFRs do not significantly impact the numbers of germinal centers or T follicular helper cells. But what we found is that the frequency of B cells that express high levels of CD138 was quite increased in the mice that didn't have TFRs. Not only the frequency, but also the numbers. So what these cells are the B cells that express high levels of CD138 are typically cells that are actively producing antibodies. So they are antibody secreting cells. So it's likely that these cells are continuously producing antibodies. This is as a lung-draining lymph node. And we wanted to confirm the results, so we use our IL-2 treatment to basically ask the same questions. We're going to deplete TFRs later on, and we're going to ask what's happened to the germinal center response. So we infect mice with influenza. Then we treat them with IL-2 from day 20 to day 30, and then we analyze what's going on with the germinal center response and the T follicular helper cell response. This is just to remind you, if we treat mice with IL-2, the TFRs are gone, or almost gone. And once again, we found that both the frequency and numbers of germinal center B cells were similar, whether you have or not TFRs, and also the frequency and numbers of T follicular helper cells were similar. But again, we have more of the CD138 positive B cells, meaning that we have more of the antibody secreting cells when TFRs were not present. So these results confirm that lack of TFRs promotes the accumulation of antibody secreting cells following influenza virus infection. So obviously, then we look for antibody titers to influenza. So we infect mice with influenza, we treat them with recombinant IL-2 to deplete TFRs, and then we analyze the influenza-specific antibody titers by ELISA. And surprisingly, we found that both mice with or without TFRs, they had exactly the same titers to influenza. And we have the same answer whether we use our recombinant IL-2 treatment or whether we analyze the BCL6 flux FOXP3 cream mice. This is surprising because we have more antibody secreting cells, but we have the same titers of antibodies. So obviously the question is, the antibody secreting cells that we are detecting, are they flu-specific? Most likely not, right? And to answer this question, we use fluorochrome-labeled recombinant nucleoprotein tetramers. So these are, the nucleoprotein is one of the internal proteins of the influenza virus. And we, in the lab, we generated tetramers with our 4 of these MP proteins conjugated to a fluorochrome. And we can use that to detect influenza-specific B cells by flow cytometry. So what we did is, again, we fed mice, we treated them with recombinant IL-2 to eliminate TFRs. And then we look into the antibody secreting cells in the control or the Treg depleted mice, TFR depleted mice. And then we ask whether these cells were flu-specific or not, using our MP tetramer. As you can see here, about 25% of the antibody secreting cells were nucleoprotein specific for influenza. And we use nucleoprotein because we know that this is the majority of the response is against nucleoprotein at this point. So 25% of cells were influenza specific. However, when you look here, despite that we have more of the antibody-secreting cells, we have only 8% that were MP-specific. So when you calculate the numbers, it comes out that in the mice that don't have TFRs, the numbers of flu-specific antibody-secreting cells are exactly the same, which makes sense because I showed you that the titers of antibodies are exactly the same. What we have is an expansion of cells that are not, or probably not flu-specific. So next question, and this is, I promise, the last question, is what is the specificity of these cells? So coming back to our Star Wars model, I told you that you have an infection with vaccines, pathogens, and tumors. The B cells in the germinal centers undergo somatic hypermutation, which is random mutation in the BCR to generate better B cells, and that's good. But throughout this process, some of these cells can potentially, you can generate autoreactive B cells through the process of random somatic hypermutation. And I told you that there might be mechanisms to prevent that. So we were wondering whether one of those mechanisms is the TFRs. So here the hypothesis is that, well, maybe TFRs accumulate later on to prevent the generation of autoreactive B cells during the process, during the germinal center reaction. So to test this idea, we infect mice with influenza. Then we treat them with IL-2 to deplete TFRs. And then we look for cell-reactive antibody-secreting cells by ELISpot. And we look specifically for B cells in the lamb-draining lymph nodes that are reacting against histone. So this is a self-antigen. And we did ELISpot. And if you're not familiar with ELISpot, every spot here indicates a B cell that is producing antibodies against histone, so cell-reactive antibodies. And as you can see here, we have very few of those in the control mice, but we have plenty of them in the recombinant IL-2 treated mice. So we have plenty of cell-reactive antibodies in the absence of TFRs. And this is true whether you use this model or we use the BCL6 FOXP3 mice. So what these results indicate is that actually TFRs prevent the expansion of cell-reactive B cell clones after influenza virus infection. So to wrap up this talk, I showed you that TFRs fail to differentiate at the peak of the influenza infection, but then accumulate later on. I also showed you that CD25 high Tregs are the precursors of TFRs, and that when the IL-2 levels decline, some of the CD25 high Tregs down-regulate CD25, down-regulate BLIM, stop to upregulate BCL6, differentiate into TFRs, and go to the B cell follicles. And in this last part of the talk, I showed you that accumulation of TFRs in the B cell follicles prevents the expansion of the cell-reactive specific B cell clones, which means that accumulation of TFRs in the B cell follicles later on is required for maintaining tolerance after infection. So I'm going to finish with the more important part of the talk, which is acknowledgements. So I want to say thanks to everybody in my lab because some way or other everybody participated in this project, but I want to especially thanks to David Ibota, who did part of the experiments, and Beatriz Leon, who is actually my wife. So I have to say thanks for many things, but also because she did experiments. And I also want to thank Uma and Scott and Holly, because we use a lot of mice for this project, and we had to generate a lot of mice, and that's pretty expensive. And these three guys were fantastic at breeding and phenotyping, genotyping, and keeping our colonies on the right size. And also, of course, our collaborators at UAB, especially to Troy Randall, who is my previous mentor, Fralin, who gave us some of the mice that that were required for this project, Amy, who gave us a lot of feedback on the possible molecular mechanism, Ken, who is always helpful for discussion, and also Warren Leonard at NIH, who gave us the IL-2 GFP reporter mice that we use to detect IL-2 producing cells. And of course, my funding sources, UAB, the CFAR at UAB, and the NIH, of course. And that's all I have. Thank you.
We certainly have time for a few questions.
So, in response to flu infection, you have IL-2.